Welcome to our video on species, community and ecosystems. This is 4.1 section B, so we're looking at the skills and applications in this video. Okay, so one of the things you need to be able to do, can you classify species as autotrophs, consumers, detrivores, saprivores, with a knowledge from their mode of nutrition? Okay, so our autotrophs, what can you remember about them? And here we've got some pictures. So is this worm, which of the types of nutrition would you say an earthworm falls as? Got some fungi there. So you need to be able to say which type of nutrition and why. Oh, a slug. A fish. Venus flytrap, there's an interesting one. This is for you to do your own research. Trees. Okay, setting up a sealed mesocosm, to try and establish sustainability. Now, you guys have set up terrariums. So we're going to see the difference between the terrarium and the mesocosm, Okay, so if you was to set up your own metacosm, these are biological systems that contain the abiotic and the biotic features of an ecosystem, but are restricted in size and are in controlled conditions. It can act as a model of a larger ecosystem in which energy enters and leaves, but matter does not. The restriction placed on a metacosm make them useful for scientific investigations, where the controlled nature of the natural ecosystem makes it difficult to collect meaningful data. Mesocosms have been used to evaluate how organisms or communities might react to the environmental change through deliberate manipulations of environmental variables such as increased temperature, carbon dioxide, or pH levels. The advantages of using a mesocosm as an experimental tool are the treatments are easily replicated. So if you're doing a trial and you're looking at something on a small scale, it's easier to replicate that and see how reliable your testing is. The effect of several environmental factors can be tested at once, whereas that's harder on a large scale. Small food webs can be established. Direct and indirect effects can be studied. And the contamination influence can be evaluated. So you're really in control of everything that's happening in that mesocosm. Constants can be controlled to see the effect of one environment factor at a time. And the sustainability of an ecosystem can really be tested. So they are miniature environments that match a large-scale environment. Now your terrarium really didn't go into that much depth. You haven't matched an actual environment that actually exists. You've made your own small terrarium. Alrighty, now we're moving on. We are looking at testing for association between two species using what we call the ChiSquared test. And this information we would obtain from some quadrat sampling. So let's have a look at what we're talking about here. Now when we do field work, we sometimes need to compare some different areas, and we can be counting numbers of an individual in an ecosystem, and we could be comparing them in a different area. But we can't just walk into a paddock and say, oh, I'm going to count all the bits of wheat in this paddock. That's unrealistic. So we need a small area that we can use as a comparison. And that small area that we use is called a quadrat. So we actually have a square that we put on the ground. It's a quadrat. And we count the different species that is in there, depending on what we're looking at. Okay, so quadrats are square sample areas that can be used to mark out our set area that we're observing. The usual procedure is to randomly position our quadrat. Okay, we might have a grid over a habitat, and random numbers can be generated to decide which grid we're going to put our quadrat into. And the area of each quadrat must be the same to ensure we've got a fair test, so a fair representation. All individuals in the quadrat must be identified and counted. If a species is unknown, then we put an X there to show that we need to identify that later. And the size of the quadrat must be consistent with the species being sampled. So in a forest habitat, you might require a bigger quadrat than you would on a rocky seashore as such. Okay, so we've got our quadrats. We've been recording the individuals within them, so the species within them. Now, to test for associations between a species, what we use is this ChiSquared test. So our quadrat sampling is a method by which organisms in a certain proportion of a habitat can be counted directly. So we can count those individuals, but how do we compare it? So chi-squared can be used to estimate population abundance, density, frequency of occurrence, distribution. Depending on what you're looking at, depends on what you're using the chi-squared test for. All right, so you can have two possible hypotheses, okay? First being the null hypothesis, two species are distributed independently. They have no relationship between the two. Or we've got two species that are associated, either positively, so they tend to occur together, or negatively, so they tend to occur apart. So we've got our two different hypotheses. To test these hypotheses, we use our statistical analysis chi-squared. Now, you can only use chi-squared if your sample size is 20 or larger. Okay, so it's important to remember that. Alrighty, so we've got a general procedure. First of all, we need to count all the individuals or estimate their percentage cover in a number of quadrats of a known size. Then to use this information to work out the abundance or the percentage cover, for example, of the whole area. Calculation. Okay, so our estimated, so here we're using the example of average density. So our estimated average density is the total number of individuals counted divided by the number of quadrats multiplied by the area of each quadrat. Write that down. Alrighty, our chi-squared test. If two species occur in the same parts of the habitat, they will tend to be found in the same quadrats. This is known as a positive correlation. If they're not found together, then it's going to be a negative correlation. Or the distribution of the two species could be completely independent of each other. Alrighty, our method for our chi-squared test. Are you ready? And you will practice this in class. First up, draw up a contingency table of observed frequencies. The numbers of quadrats containing or not containing the two species. So we've got species A present, species A absent, species B present, species B absent. So here, both species A and species B would be present. Here, species A would be absent, but species B would be present. You need to ensure you fill in your totals, okay? So calculate the row and column totals, add in together what's in the rows and columns. Okay, now you need to calculate the numbers of degrees of freedom using this equation. So M is your number of rows and N is your number of columns in your contingency table. Find the critical region for chi-squared from a table of chi-squared values. You'd be given your table of values if you was doing this in a real life situation. So these will always be provided with you using the degrees of freedom you have calculated and a significant level probability of 5%, okay? The critical region is any value of chi-squared larger than the value in the table. So, next up, and that should be a number 5, but I couldn't get that to happen. Next up, we need to calculate our chi-square using this calculation. Okay, next step, calculate chi-squared using our calculation. So that's the calculation we just had. Okay, and here it is, just looking a different way. This means you will need to work out 4 observations minus your expected value, okay, and then squared, and then find the sum of them. Okay. You then need to compare the calculated value of chi-squared with the critical region. The critical region is any value of chi-squared larger than the value in the table. If the calculated value is in the critical region, then there is evidence that the probability is much lower than 5% value, which is generally regarded as significant, and the null hypothesis is then rejected. This means there is less than 5% probability that the 2 species are together or apart by chance. If the calculated value is not in the critical region because it is equal to or below the value obtained in the table of chi-squared values, then your null hypothesis is not rejected. There is no evidence at the 5% level for an association between the 2 species. It is a probability of 5% or greater that their association is due to chance. So therefore, a value considered significant if there is less than a 5% probability. The results are attributed to chance. And there we go, we've finished our application and skill introduction, but of course that chi-squared we need to practice in class a lot further. It's just your introduction on here. See you in the next video.